This breakthrough genomic test can diagnose nearly any kind of infection, and it can do that much faster than any other test that has previously been used. So this comes from the University of California at San Francisco, where scientists have been able to develop a next generation metagenomic sequencing test that can rapidly and accurately diagnose a wide range of pathogens. And by pathogens, what we're talking about here is viruses, bacteria, fungi, parasites, all sorts of tiny little creepy crawlies you can get in your body. They can do a lot of bad things. Trust me, I know I got some of these things once. And what's interesting about them is that they were able to speed it up by, instead of designing a test that tests for one pathogen at a time, and typically you would do that in a sequence, be like, yeah, it's not this one, yeah, it's not this one, yeah, it's not this one. Instead, their new metagenomic testing analyzes all of the nucleic acids, all of the RNA, and all of the DNA all at once, all in a single sample. It's almost like a brute force method. As the research lead doctor says, our technology is deceptively simple. By replacing multiple tests with a single test, we can take the lengthy guesswork out of diagnosing and treating infections. And what's wild about this test is that they're actually analyzing cerebrospinal fluid, which, as the test points out, is a pristine liquid that bathes the brain and spinal cord. And what's useful about this is if the liquid is pristine, it's probably a lot easier for the testing apparatus to rapidly identify and sequence the genomes of anything in that pristine liquid that's not supposed to be there. If you were to do this with blood or like a random tissue sample, your results would probably be a lot more chaotic. But what's also cool is that this test takes just about 48 hours to reveal results. They've already done this on over 5,000 people. Over the last, I don't know, decade or so, a little bit less than a decade. And the test was able to accurately identify the pathogen 86% of the time, which is extremely good for an experimental test and will only get better as it deploys into public use.